if you take a bunch of human neurons and you grow them on a lawn on a petri dish, and then you take a scalpel and you put a big scratch through it, so like a wound, through a neural network on a dish. You put the anthrobots in, they sort of run around. Some of them will find the wound. They settle at a particular location in the wound. They form this larger structure we call a superbot cluster, which might have, 8 to 10 bots. And 4 days later, you lift them up and you see what they were doing while they were sitting there is they're knitting the two sides together. So they were actually healing the gap. Like we could have, who would have known that your tracheal cells, right? It's absolutely amazing. Who would have known that your tracheal cells that sit there quietly in your airway dealing with, pollen and whatnot, have the ability to run around and heal neural cells? We could not have predicted that. What I have claimed about consciousness in general is that for the exact same reasons you attribute consciousness to some other beings, you should be very serious about attributing it about the potential for it to exist in other organs and tissues of your body for the exact same reasons. I think all living material and probably lots of non-living material has some kind of experience, but being able to say what that is requires a lot of work that hasn't been done yet.
Do you think we need different words for describing cellular cognition compared to human cognition, or do you think they exist? They should exist rightfully on the same continuum.
Well, I absolutely think they exist on the same continuum. And the way we know that is because we can take concepts that were traditionally used for behavioral and cognitive neuroscience, and we can use those same exact concepts profitably in other sciences. So I'll give you an example. And I used to have my students do this by hand, and now we've actually made a tool. I could give you a link online. There's an online tool that does it for you now. You take any neuroscience paper and you just do a find or replace. Okay. And so anytime it says neuron, you say cell. Anytime it says millisecond, you say minute. And then you can do some other pivots because instead of three-dimensional space, you're talking about anatomical amorphous space. But you make those changes. That's it. Everything else fits. You got yourself a developmental biology paper, because all of the concepts. I mean, we've used it all. The tools, optogenetics, neurotransmitter modulation, ion channels, electrical synapses, the concepts, perceptual bistability, illusions, memory, active inference. You name it. We've used all of this stuff in morphogenesis. Because and it works because our cognition is homologous, not just similar to or not just analogous to, but actually homologous to the morphogenetic intelligence. That is where it evolved from. All of the things, all of the tricks that the brain uses to do counterfactuals, to have memory, to do goal-directed behavior, all of these are ancient. They predate neurons by a really long time.
Right.
And what evolution did was basically speed up electrical and neurotransmitter processes, and then it pivoted them from the original space they operated in, which was anatomical space. The first use case for this was to navigate anatomical space, and then it got pivoted to when nerve and muscle appear to kind of run around this three-dimensional space. So all of the concepts transfer. Now, that doesn't mean that we're not going to have to broaden some of them, and there will be some aspects of memory, for example, in gene regulatory networks that aren't exactly like all the kinds of memory that we study in psychology, right? So there will be some, you'll need to make some tweaks, but overall, it's exactly the same. But again, the bigger, so the practical point is that it's absolutely a continuum, and the way you know it's a continuum is that you can move concepts back and forth. People often say to me, it's confusing when you use the same terms. You should come up with different terms, and then you won't have to fight with anybody. So we do memory, and then the cells do shmemory. And it's fine. Those are different things, and you can, right? But the problem with that is that it's actually it's a barrier to new discovery. Because the whole point is that it then tells you, pretending that these are different categories keeps you from porting tools and concepts. And for the same reason that Newton wouldn't have been wise to say, well, gravity is the thing that makes the apple fall, but schmavity for the moon, then we'll have these two different things, right? For exactly the same reason that would have been a mistake. You don't want to make up new names for this unless you actually find out that the tools don't work, and then it's a different phenomenon. But the other important philosophical point here is that I don't believe it's the case that everything is one thing And our goal is to discover what it really is, and we can fight about it for a while, but then eventually we'll sort of figure out what it really is, and then we can move on. Philosophically, I think that's the wrong way to think about this. I think that the spectrum of cognition, or as I call it, the spectrum of persuadability, is what when you say this system has a particular degree of intelligence or a degree of cognition, what I think you're really saying is here what you're talking about are interaction protocols. This is a very engineering, I mean, I'm an engineer, it's a very engineering perspective. What you're really saying is, here is the bag of tools I plan to use with this thing. Maybe it's hardware rewiring, maybe it's changing set points from control theory and cybernetics, maybe it's training from behavior theory, maybe it's psychoanalysis, the friendship, love, whatever it's going to be. Here is the set of tools I'm going to use to interact with the system. Watch how well I do, and then we'll all see how well it works out. And so what this means is that there isn't one thing that anything is. I mean, all of these terms, they're metaphors, they're mental models. They're not, nothing is anything in the sense of these things. Nothing is a computer, nothing is a machine or an organism. All these are all metaphors that bring a set of tools with them. And so it's perfectly possible to have one object, a cell, you can look at a Zenobot and you can say that is a biological robot. Or we can say, no, that is a proto-organism. Or we can say, no, that is a set of cognitive propensities, or that is a discovery tool for regenerative medicine, or it's right. So depending on, I'm going to use it like an organoid to find to screen drugs for regenerative medicine. No, I'm going to interact with it on a cognitive level and see what it knows, and I'm going to treat it as a proto-organism. Or no, I'm going to make I'm going to use it like a biorobotics platform and have it clean up the waterways. All of these are valid. It's not what it really is. It's what do you see in that system, and what bag of tools can you use, and how did that work out for you? It's a very sort of experimental kind of thing.
Right. It's so well said. I think it's so well explained. I think biology runs into a lot of traps with language, because language, by nature of language, is discrete, right? Here's a box, here's a category, here's a box, here's a category. But the more I learn about biology, and how you so eloquently put it there, is that everything exists on spectrums and continuums, and language is just kind of tricking our brain into thinking that these things fit into discrete boxes. It's the same with psychiatric conditions and diagnoses, like autism. These things are spectrums of phenotypic behaviors, but everything is far more complex than these niche labeled boxes. And there's something about the brain that also really likes categorization and chunking as a way of memorizing and learning fields. We think in discrete boxes, but it tricks us in a lot of ways. It's hard to get out of that, hard to, I think of things entirely in continuums and spectrums, when language has been kind of tricking us our whole life into thinking they're discrete boxes.
Yeah, it's and it's not just biology. It's everywhere. Regular people who are not biologists or anything will always tell you, "Well, I know the difference. There's live and there's dead. There's living and there's machine. There's true minds, and then there's faking it with tricks. These things." They are also an important frame. These categorizations are an important frame, and the way that I explain it is like the word adult. Okay, so we have the word adult because in the court system and social policies, it lubricates that whole thing by saying, hey, look, on the night of your 18th birthday, bang, you're now an adult. Okay, and all that does, it's a very expedient, practical thing, and if you want to rent a car, it's actually 25, and with or whatever it is, and so you make up these things, but let's not then pretend that this is real. We know nothing happens on the night of your 18th birthday, and if you really want to talk about what an adult is, you have to fundamentally unpack the nature of personal responsibility and metamorphosis and maturation and change. And are you the same person you used to be because you grew up, or because you had a brain tumor, or a neurotransmitter insufficiency? And how did you get here from being a single cell in embryogenesis anyway? At what point did you acquire this magical property of being responsible for your actions? What could that possibly even mean, right? So all these questions, you don't want to address all of these questions every time you need to register to vote or whatever, right? And just, oh my God, what really is a human responsibility? So we make up these easy categories, live and dead, or like all of these things. But when you dig into these things, you find out that if you really want to understand what's going on, to really get at the nature of it, what you want is not some sort of binary category, which inevitably there will be crazy corner cases on things that we don't know how to categorize and whatever. What you really want is to understand the process of transformation. what is actually happening to increase your adulthood? How did you, because there was no magic lightning bolt that made it happen. What are the processes that increase it from the time you're a cell to the time that you're a modern human, adult human? So these things, it's really important that these categories, to remember that these categories that we make up, they're just convenient metaphors that we use to make life simple. They obscure the underlying continuous nature of things. I have debates with people all the time where they try to come up with examples and say, okay, well, here, how about this? Surely this is a binary category. And then I sort of zoom in and I show them how, no, actually, here's the in-between case. I can only think of one example. I'm not even sure I believe that one, where I think it really is, where it might be binary, and that's certain things in quantum mechanics. But otherwise, pretty much everything that we care about is a continuous spectrum. And the far more interesting story is the movement across that spectrum. It's not trying to maintain these categories.
Right. I'm really curious about the development of when the Zenobot is clumping its pile of cells together. And you speak of everything as a continuum. That clump of cells is getting bigger and bigger. At what point does it start exhibiting the behaviors of the Zenobot that is creating it? Does it have movement right away? Is it able to repair itself? Do you see what I mean?
Yeah.
Like, that's a really complicated idea for me. That does it, if it's all a continuum, is it at the level of one cell already exhibiting some of the properties of the whole parent Zenobot? And then as it gets more complex, as more cells are added and added and added, it starts to look more like its buddy that it's being created by. Have you charted that sort of relationship? How as that clump of cell gets bigger and bigger, sure, and sort of how it gets more intelligent, and speak around that a bit, because that was kind of breaking my brain trying to think about how that relationship works.
Yeah, we absolutely have, because one of the beautiful things about these kinds of models is that they're right there in front of you on the benchtop. You can record the whole thing. It is definitely a journey, because the initial bot, it does not move. It's immotile, and then eventually it does move. But the transition, of course, is slow and gradual, because the way that it starts to move is it grows these cilia, right? So these little waving hairs that come out of that grow out of every cell that row. They basically row against the water, which is how this thing propels forward. So think about what happens. You have to be covered in, I don't know, thousands and thousands of cilia. Well, how many do you have at any given moment is a question. And once you start moving, it's kind of like a baby. Do babies move through the world? I mean, kind of, but better at 6 months than at 1 month, and better in a couple of years. So same thing. They sort of start wiggling, and then they sort of start, and then eventually the cilia align so that they can row in opposite directions, and the thing actually moves forward. So yeah, you can watch that developmental process. It is slow and gradual, like all developmental biology. It's everything is slow and gradual. And then it acquires, and some of these bots are better at, if you say, you can't say, can it do kinematic replication? The question is how well, because we can make a whole spectrum of ones that are really good at it. We can make ones that are not as good at it. And that's what you see. You see as they develop, first they're not that good at it. Then eventually it really cranks up. They do great. And then eventually a few days later, they sort of degrade and start to die. And then it stops again. So yeah, continuum all the way.
And does it make exact copies of itself, or rough copies of itself with errors?
Good question. This is not what I would call strong heredity, in the sense that when they make the thing about standard reproduction is that what happens is that the offspring that you make are more similar to the parents than they are to random members of the population. Yeah, so there's some sort of you pass on whatever traits you have. At this point in Xenobots, we don't have any evidence for that. We don't know that it's not doing that, but we don't have any evidence for it. They basically, all the bots make standard bots. There is a bunch of variability there. They're not all identical. There is a range of parameters in terms of the shape, the speed with which they move, the different kinds of behaviors that they do, and so on. There's a spectrum, and we don't have any evidence that a particular bot that makes another bot, that one would be more similar to its parent than to any other bot that was made in that same dish. So it's sort of again on the spectrum. Like, is it reproduction? Well, it's missing some of the features, but it certainly has other features. So It's not whether it is or it isn't. It's what are the metaphors that let you move forward in discovery.
Why did you even think to sprinkle those cells in the dish? Is that one of the products of the computer algorithm that said it might do this if you sprinkle its component parts sort of around it?
That AI didn't have the ability to predict to suggest new experiments like that. That was something that Josh and Doug and Sam and I thought about. We watched the movement, and one of the things that we studied early on is to sprinkle a bunch of passive particles in there, because the particles let you see the movement of the water as the cilia are beating. It's really cool. You can these tiny little particles that reflect light, and you can sort of see all the vortices and everything else that's happening in the water as this thing progresses. But watching it displace those particles and watching them collect in little piles and so on, it became obvious that these things exert force on the environment around them. And we wondered, okay, we can see what they do with a passive material. What would they do with an agential material that, once you do collect it into piles, isn't just going to sit there as a pile? It's going to do what cells do, which is to try to create something bigger. And some of the videos, I can send you a link to some of these things, are absolutely incredible. When you watch what the cells are doing as they're being gathered into a pile, there are so many levels of life here. There's the collective, so there's a population, and there's 20 bots doing this dance, and they're all moving when they're interacting with each other, and they all of that goes into redistributing the cells. Then there's the individual bots, and the individual bots interacting with the cells and with the water. And then there are the cells themselves. And you can watch as they're being placed into these piles, what are the cells doing and how do they start to come together. And then, of course, inside the cells are the molecular networks, which now we can also read out via calcium signaling. So we have a couple papers coming out looking at taking techniques that are normally used to analyze brain function, so calcium imaging, right, is in neural systems, and we've applied that to Zenobots. And there's some amazing properties. There's some amazing properties, and in fact, we compare them head to head. Here are things like various statistical and causal geometry kinds of metrics of what happens in the nervous system under different kinds of cognition, and then here's what happens in Xenobots. So you can see it head to head what the differences and similarities are.
That's so cool. Is there anything specific? I presume there isn't something magical about the Xenopus frog. If you took the exact configuration of cells, but substituted them all for the albino silverback gorilla, random example, no idea why I went there, but would you be able to make any predictions of what that system would do? Would it act in a completely different way? Is there anything you can predict about that system?
Well, a couple things. First of all, no, I don't believe there's anything unique about the frog system. We did it because it's very tractable and easy to use and all of that stuff. In particular, though, there's an issue kind of like what we discussed at the beginning, which is this: if we had done it with a set of cells that don't have cilia, they wouldn't be moving around in the three-dimensional world, and they might be doing all kinds of cool things. They might be moving through transcriptional space and physiological space. And we could and do chart all of that. And I will say to people, look, this is behavior. This thing is navigating space. It's avoiding local barriers. It's got delayed gratification, whatever it's doing. And people will say, that's not behavior. That's something else. Let's call it physiology. And they'll say, yeah, but we're using all the tools of training and, visual illusions and all this stuff and so on. That's the fight that you have to have. But when the thing is moving in three-dimensional space, it's easier for people to see what's going on. And so that's why, that's why we started with ciliated cells, because you don't want to fight every battle at once. You want to be able to say, like, this really is behavior. We can all agree it's behavior. Now let's talk about what that means in non-neural cells. But we also study a bunch of other things that are not moving in 3D space. And I frankly think that the vast majority of organoids that people make that don't move, that just sit there in a dish, I think they have the equivalent of locked-in syndrome, meaning that there's tons of processes going on. They're solving all kinds of problems. They're communicating to us. They're doing all sorts of interesting things. We are blind to it just because we're so fixated on the three-dimensional world. So for that reason, we chose ciliated cells. Now, with respect to making this out of other animals, let's talk about anthrobots for a second. So when we made these Xenobots, one of the things that some people said was, Okay, well, they're amphibian cells, and we know amphibian cells are pretty plastic. They're embryonic cells, and we know embryonic cells are plastic. So maybe this is some sort of a frog specific thing. It's a weird quirk of frog developmental biology. People have been using animal caps, which is where these cells come from, for a long time. People have seen them wiggle around. Nobody's kind of made anything of it, but that's been seen for a long time. So maybe this is just some aspect of frog developmental biology. It's like this weird fluke one off. And so at that point I thought, okay, let's get as far away from embryonic frog that we can. And what would that be? Well, an adult human. And say, fine, let's go to an adult human. Nothing to do with frogs, nothing to do with embryos, nothing to do with amphibian morphogenesis or animal caps or any of that. Let's see what adult human cells do. And so this is Gizem Gamushkaya, who got her PhD in my lab recently, developed a protocol for basically taking tracheal epithelial cells donated by human donors. Some of them may or may not be alive after this. So it's a weird kind of, as these things are running around our lab, it's a weird kind of life after death for some of these patients. Right. But what we found is something very interesting. First of all, they do something similar to what Zenobots do, meaning they self-assemble. They make this basic, simple architecture where it's basically a spherical thing with cilia on the outside that then runs around. So that's similar. Instead of hundreds of altered gene expressions, they have thousands, about 9,000. So about half the genome is radically differently expressed. They have, similar to the Zenobots, they have a small set of discrete behaviors, and you can draw an ethogram of the transition probabilities between the different behaviors, just like you would for any animal, fish or whatever. They have all sorts of interesting capacities. So let's take stock. Could we have predicted the fact that this would work with other species? We could, and we did. I did predict that, and we did in fact find that. Could you predict what the shape of it was going to be and what the behaviors are going to be? Sort of. You could have predicted that it would be similar, but actually that's not a super strong prediction because there are other differences that we couldn't have predicted. Could we have said which genes were going to be altered? We could not, and that's because what I think is going on here is that as we see, they say, well, it's the genome drives, right? Because dogs have puppies and cats have kittens, and it's the genome that drives. Well, yeah, but the human genome also makes Zenobots, and the anthrobots, and the frog genome makes Zenobots, and so on. It's the genome has a set of affordances that the cells can exploit. So they can express different genes because of a new lifestyle. I mean, think about what's happened here. There are no scaffolds. There's no genomic editing. There's no synthetic biology circuits being knocked in. There are no weird drugs or nanomaterials. There's none of it. It's just a different lifestyle. You're living in the same environment you would have been living in anyway, but the other cells aren't there to tell you what to do. So you're liberated in a way, right? The normal, for example, in Zenobots, those epithelial cells would normally be sort of bullied by the other cells into being this boring two-dimensional outer surface to keep out the bacteria. In the absence of that, they do whatever they want to do, which is this. And so how much of that could we have predicted? So we certainly predicted that it would happen, but the specifics of it, no way. We had no idea which genes they were going to turn on, or the fact that, for example, anthrobots, if you take a bunch of human neurons and you grow them on a lawn on a petri dish, and then you take a scalpel and you put a big scratch through it, so like a wound, through a neural network on a dish. You put the anthrobots in, they sort of run around. Some of them will find the wound. They settle at a particular location in the wound. They form this larger structure we call a superbot cluster, which might have 8 to 10 bots. And 4 days later, you lift them up and you see what they were doing while they were sitting there is they knitting the two sides together. So they were actually healing the gap. Like we could have who would have known that your tracheal cells, right? It's absolutely amazing. Who would have known that your tracheal cells that sit there quietly in your airway dealing with pollen and whatnot have the ability to run around and heal neural cells? We could not. We could not have predicted that. And something else that's important is that ability was not experiment 82 out of 1,000 things we checked. That was the first thing we tried. That was the first damn thing we tried. And so what does that tell me? Well, that tells me that they probably do 1,000 other things that we just don't know about, that we don't, we're not smart enough yet to chat. I mean, we're now going through and looking for all kinds of other things, but that was just, it was just a total, total luck. But it's not a needle in a haystack. They have no doubt, they have tons of these capacities. And the question is, and I think this is really important. Anytime you try to establish or claim the cognitive capacity of a system, it's a two-way IQ test. What you're really doing is taking an IQ test yourself. What you're saying is, how smart was I to be able to determine what space is it working in, what are its goals, what capacities does it have to meet those goals, and design experiments to show that. All this is, it's a reflection of our own largely inability to deal with minds that are radically different from our own. So that's the tools that we're trying to make now.
It's amazing. It's amazing. It really does feel like it's conscious. And I'm going to define what I'm considering conscious here so that everyone is on the same page, where it seems like they have subjective experience. There's some experience of what it is like to be the Zenobot or the arthropod. It's sensing the environment, it's reacting, it's integrating its signal, it's responding, it's learning in some ways. As you can say, there's ways to communicate it. Like, for me, at least, that absolutely passes the bar of if this thing is conscious.
So let me say a couple of things about that. First of all, just to be clear, I have not claimed, and I don't think anybody on our team has claimed anything about consciousness for these things for a couple of reasons. That's hearing those reasons. So we haven't. I haven't said they're not. Because again, I don't think you can just sort of sit back and have opinions on this. You have to do experiments. But I haven't said one way or the other. And but I can say a couple of things. Consciousness is obviously a very difficult issue. We're not going to resolve it in the next 15 minutes. But there are two things. There are two things here to think about. The first is that typically there are three kinds of reasons that people that people use to attribute consciousness to others. So this is the other minds problem, right? So you and I presumably think that each other is conscious. Now, why would that be? We don't have direct access to each other's minds. Why do I think you're conscious? So people typically like three reasons. One reason is behavior. It's all the things that you just said. Does it have goal-directed behavior? Does it seem to have valence about what it, preferences about what it's going to do? Does it have learning? All those. So publicly observable behaviors. Now, okay, and so that's one, and some people don't like those because they'll say, well, a guided munitions like a self-homing bomb has all of these things. It's got goal-directed behavior, and some of them can learn from experience and blah blah blah. Right? So some people don't like the behavioral, but other people do. Then you've got the architecture argument. So it's like, look, I know I'm conscious. I know what I'm made of, presumably, and if you're made of the same stuff, then probably you're the same. So if we look at that, so what are we made of? We've got some ion channels, we've got some gap junctions, we've got some neurotransmitter pathways. All of the organs in your body, all of the systems in your body are continuous with that. You have ion, you have all that machinery exists in every cell in your body. There are electrical networks throughout your body that make decisions about morphogenesis. So what I have claimed about all of this. So we've got behavior, we've got composition, and then we've got evolutionary lineage. Right. So you and I come from the same path on the evolutionary tree. So if I think I'm conscious, then I'm going to say probably you are too. So using any of those criteria, and I'm not aware of really any other. I mean, there's things like IIT where they look at information, like causal architecture in the information structure and so on. But typically, what I have claimed about consciousness in general is that for the exact same reasons you attribute consciousness to some other beings, you should be very serious about attributing it about the potential for it to exist in other organs and tissues of your body for the exact same reasons. The fact that you don't have access to it doesn't mean anything. In other words, I mean, my left hemisphere doesn't have access to your consciousness either. So of course I don't feel my liver being conscious, but we should be open to that possibility because that material does all the same behaviors in different spaces, but it does all the same behaviors. It is in evolutionary continuity and it has all the same composition, it has the same mechanisms. So we should be open to that. However, what that doesn't address is is what they call the hard problem of what is it actually like to be that creature. And if I absolutely think that Zenobots have an inner perspective. I don't claim it. We don't have any data on it. It's very hard to do these things that we can't even really strongly do that for each other. But as much as we can, my suspicion is that, of course, they do. But then again, I think that very widely across the universe, extremely wide. That experience is actually extremely widely distributed. Some degree of it is extremely widely distributed across the universe. But what I can't do is tell you what kind of what is it like to be a Zenobot? What kind of consciousness does it have? What is it actually aware of? What is the valence? We don't have those data yet. When we do have those data, we will be able to have the same kind of discussion that we currently have about lots of different animals, insects. Insects. For example, one of the things that people talk about in bees and so on is self-medication. They'll say that insects will, they'll if they lose a limb or something, they will try to either try to. So sometimes certain animals will seek out analgesics in the environment. Others will sort of groom it. They show that they're aware of the thing. They show that there's so we can have those same kind of discussions. That still doesn't get you across the so-called hard problem, but for sure we can have those same discussions. There will be an additional twist to it, because again, it'll go beyond three-dimensional behavior. So if you're a being that lives and navigates and navigates an entirely different space, let's say the space of physiological states or anatomical morphospace instead of the three-dimensional world. But that is where you have your perception action loop. You take sensor readings, you have memories, you make decisions, you have beliefs about priors about what happened before, you have goals you're trying to reach, you have ingenuity about getting around that space, all of that. What is it like what is the consciousness like of a being that inhabits one of these other worlds, And I think it's there. I personally absolutely think it's there. But being able to say something about it that's useful is an entirely different matter. So I think we have to be careful. I'm of course very open, because I think all living material and probably lots of non-living material has some kind of experience. But being able to say what that is requires a lot of work that hasn't been done yet.
Giant news. We created a completely free ebook titled 26 Neuroscience Books for 2026. I've read over 100 neuroscience books in my life, and with Christmas just around the corner, I thought I would help people find the perfect book for them or to buy for someone in their life. It includes really nice science-based illustrations, an accessibility guide, and personally written segments for each book and the impression they left on me. I also asked 100 top scientists like Dr. Mike Levin, Anil Seth, Nick Lane, and many more what their favorite science book is, and you can find all of those included at the end. If you want to support the channel, you can also purchase a beautiful physical paperback copy, also a perfect stocking filler. We put a lot of effort into our first Giant Shoulder product, and we cannot wait to hear your thoughts. Check it out in the link below. Again, it's completely free.